When I tell people about the research that I'm doing, they don't really understand it at all. They don't know what the word ectomycorrhizal is, and I'm sure most people don't. I think mushrooms are beautiful, underappreciated, and they're like everywhere, and people don't see them. A day at the Arboretum, I usually start by going on a walk around in a route. I have a designated route that I go on that passes all of the ectomycorrhizal trees. The types of mushrooms that I've been finding around here are generally ectomycorrhizal because those are the ones that actually produce a fruiting body. Ectomycorrhizal fungi grow in association with different trees, and the species of tree actually really matters on what type of fungus it associates with. So manzanitas associate with one type, and like oaks would associate with another. So when I'm in the field, I take a photo and I upload it to iNaturalist, and it helps me identify it, and also it creates a place where this identification has been made, and I can link that to my database. So we've just stumbled across an Inoscibe species. These are mycorrhizal mushrooms, and they actually associate with oaks. This is a remarkably hard to identify species, just because they all look the same. Generally, you need spore prints and microscopy to identify them. So we can't do that in the field right now. But it's important to this project because it's in mycorrhizal association with this oak, and it actually associates with a bunch of other oaks that are nearby. So it forms a kind of like a web of all of the oaks connecting them together. Once I collect the mushrooms, I dry them in a dehydrator, and then I am able to send the actual dried fungus to a lab, and they're gonna do the sequences for me. The sequences are gonna allow me to identify the mushrooms more than I can. So a lot of the mushrooms that I've been finding, I can only identify down to genus level because species can sometimes be really hard to identify, and also because some of the species that I'm finding here might not be native and might not be named. No one at Santa Cruz has done the work that I've been doing here, especially at the Arboretum. There's no mycofloras that have been done specifically to get a full collection of what grows in the Arboretum. Kind of surprising just considering that Santa Cruz is fungus friendly. I grew up going on mushroom walks with my parents, which I'm very grateful for because it made me not afraid of mushrooms. I kind of forgot about those mushroom walks when I was a kid, and I became interested when I started finding them on my own, and I really started developing an interest myself. I think that this work could lead to a senior thesis for me. We don't know a lot about introducing non-native fungi, and we don't know a lot about what is native and what is not native. Just because there's no historical baselines for what grows here. So that's why mycoflora is so important to start with, is because you need to have an understanding of what is here right now. The final part of this internship and research project is going to be a field guide, and ideally you're going to be able to take a walk in the Arboretum and find things that you can see in the field guide. And another part of the research is to find associations with the trees that grow here. And there's a lot of non-native trees, and I'm interested to see if those non-native trees have brought non-native fungi with them through their ectomycorrhizal associations. Non-native fungi can be an issue, and I think that one of the biggest examples of that is death caps. So death caps were introduced from Europe, and they were brought in as a mycorrhizal fungus through oaks, and they are very invasive and have spread across California and many other states, and they've killed people. This is a great way for me to actually go out, look for mushrooms, which is what I would be doing anyways, and have a purpose behind it and like actually produce something that's valuable.